Hello everyone, and welcome to Assessing Differential Expressions of Protein Interactions in RNA by Combining isPLA and HCR pro-RNA fish on BondRx Automated Research Stainer. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational webinar is presented by LabRoots and brought to you by Leica Biosystems, Molecular Instruments, and DaVinci. To learn more about our sponsors, please visit the links there on the registration site. We do encourage you to participate by submitting your questions during the presentation or after the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time at the end of the presentation. You can also submit any technical issues there as well if you're having trouble seeing or hearing the presentation. I'd now like to welcome our first speaker, Kendra Ritchie, Field Application Specialist, Life Sciences at Leica Biosystems. For a complete biography of any of our speakers, click on the biography window on the left side of your screen. Kendra, you can now begin your presentation.
Good morning, everyone, and thank you for joining us today for this webinar, Assessing Differential Expression of Protein Interactions and RNA by Combining In Situ Proximity Ligation Assay, or isPLA, and HCR pro-RNA fish on the BondRx Automated Research Stainer. I'm Kendra Ritchie with the Life Science Field Applications team at Leica Bio, supporting the BondRx and RxM staining platforms. If you're not familiar with the Bond research stainers, the BondRx and RxM, they provide a customizable staining platform and full automation, including full automation of Molecular Instruments HCR Pro RNA histotechnology and DaVinci's isPLA technology. By providing superior quality and flexibility while enabling the automation of IHC-ish and emerging tests, the BondRx research stainers help researchers drive their biomarker discoveries forward. Today, we're going to discuss how in 3 ways. The first being by exploring your ideas, learn about tests you can run on the BondRx using its open and customizable system. 2, accelerating your test program. Explore how the BondRx accelerates your test program. And finally, commercializing your discovery. The BondRx provides a clear path from research to clinical. Moving into exploring your ideas, also here on the screen, you can see a beautiful image of an HCR Pro RNA fish stain by Molecular Instruments combined with an IF membrane stain. The flexibility offered by the BondRx system allows you to discover technology such as the HCR Pro RNA-ish, isPLA, multiplexing, and more. So on this slide, you can see some representative images of staining that's been performed on the BondRx using both DaVinci and Molecular Instruments assays. If you look at the top right image, you see MI's HCR Pro RNA-ish plus IHC on tonsil. And at the bottom left, that image displays DaVinci's isPLA on colon using the DaViniflex kit. Here's where the customization abilities of the BondRx really come into play. The open software allows researchers to design their own unique and ideal protocols to fit their specific needs. So customization can be performed across all editing segments of the protocol. When we're looking at pre-staining protocol segments, you can customize preparation and antigen retrieval. You're able to customize bake and dewax options, enable one or both, disable both if you are working with frozen tissue. You can adjust the amount of dewax steps you would like, as well as baking conditions. You can also customize antigen retrieval, probe application, and removal. You can adjust incubation times and temperatures for retrieval. You can also disable retrieval altogether if preferred. In the staining protocol segments, you can customize any step within the protocol. This allows for you to use your own preferred markers in open bond containers, create your own detection kits for your specific assay needs. You're able to select from 3 available dispense types, each with a specific function for preserving stain and tissue quality, and change incubation times and temperatures on any protocols. You also have the ability to insert additional wash steps or edit current wash steps within the protocol. So as you can see, everything's extremely flexible and customizable within protocol edits. Accelerating your test program. Features of the Bond RX are designed to enhance the user experience and achieve research goals efficiently and effectively. One feature is flexible test management. The antibodies, probes, and detection systems can be accessed at any time during a protocol when the reagent is not actively being used. Reagents can be placed in any 9 positions over 4 trays for a total of 36 positions for individual reagents or detection systems. For bulk reagents, minimal maintenance is required. You have the capabilities to fill bulk reagents during the runs without affecting the protocol, and the BondRx overall generates low volumes of waste. The BondRx also has a small footprint within the lab to accommodate for space constraints. Maintaining the integrity and morphology of tissue is crucial within research. The BondRx utilizes cover tile technology to protect the tissue, as well as provide reproducible and consistent staining. The main aspirating probe gently dispenses the reagent. The cover tile technology then employs capillary action to disperse the reagent underneath the cover tile and uniformly cover the tissue. This provides excellent tissue retention, morphological preservation, and complete and even reagent coverage. The cover tile system also provides a controlled incubation chamber, allowing the tissue to stay hydrated, preventing any reagent loss during staining, especially on crucial heated steps or longer incubation times. The cover tiles are also easy to use with keyed placement, so they cannot be loaded incorrectly, and they have an easy maintenance schedule. That is kind of a high-level overview of the bond and its functions and capabilities. These allow us to collaborate with partners such as NaVinci and Molecular Instruments to streamline your tissue-based research needs. These partnerships are available on the BondRx as preloaded reagents and protocols with the following BXD version updates. The NaViniBrite chromogenic isPLA kits were automated and launched on the BondRx and RxM with BXD version 38 in June of 2024. The NaViniFlex fluorescent isPLA kits were automated and launched on the BondRx and RxM with BXD version 40 in November of 2024. And most recently, the HCR Pro RNA-ish assay was automated on the Bond in January of 2025 and will be available with BXD version 41. Information for updating to these BXD versions from your current BXD version can be obtained from either your local Leica field applications specialist, as well as instructions listed in the BondRx IFU for how to update. And these BXD updates exist on the Leica Life Sciences website. Joseph at Molecular Instruments and Sara with NaVinci will go into greater detail about each of their technologies. But before I hand off, I wanted to touch on what those technologies look like on the BondRx and how automation is beneficial. With NaVinci, the BondRx gives you the capability to automate detection of protein-to-protein interactions and post-translational modifications through isPLA, reduce hands-on time using preloaded staining protocols for chromogenic and fluorescent detection, and the capability to stain up to 30 slides in 9.5 to 12 hours on the BondRx and RxM systems. With molecular instruments, you are able to detect a wide variety of RNA targets while preserving tissue morphology without any digestive enzyme treatment, utilize preloaded staining protocols for HRP and AP detection, easily modify protocol with instructions in the setup guide for fluorescent detection, and stain up to 30 slides in 7.5 to 11 hours on the Bond RX and RXM. And that is it for me. Thank you all again for joining today, and I will hand off to Joseph at Molecular Instruments.
Thank you, Kendra. My name is Joseph. I'm on the business development team here at Molecular Instruments. I'll go through some background on our company and how you can use HCR Pro for your needs. Molecular Instruments is the company itself, and HCR stands for hybridization chain reaction, and it is our platform technology that we have built our RNA-ish kits for. So some background to our company. So in 2010 is when we conceived of the HCR platform. It was developed by our co-founders at Caltech. During this time, it was being used for a lot of developmental biology uses, both with other collaborators at Caltech, but also other academic institutions across the country. In 2018, we fully spun out as our own independent entity, still developing kits for our DevBio users and other academic collaborators across the country. In 2023, though, is when we officially launched our kits for use on autostainers like the Leica Bond RX, targeting our biopharma users. And in 2024, we actually have our first approved LDT. So some features of the HCR platform for use with those academic users, they are used with a manual protocol, and they are used under our HCR Gold brand. But we'll be focusing on our HCR Pro line. So these kits are designed for use on autostainers like the Leica Bond RX and offer a number of features for use cases with that system. So this is a nice little image. This is actually from one of our academic collaborators using the manual protocol, but it is a whole mount embryo and speaks to the flexibility and ease of use of the platform. It's very compatible with a wide, diverse range of sample types. And as you can see here, it is able to be used with more trickier sample types like a whole mount embryo. So as for the HCR platform itself, this is a bit of how this amplification system works. So we begin with these I1 strands on the bottom left. So these are actually conjugated to the RNA probes themselves. These probes are designed in-house for our users. All the user has to do is provide us with the target sequences of their targets, and we conduct the probe design and synthesis all in-house. Once complete, these are conjugated to these initiator strands. And when the probe hybridization step is complete on your Leica Bond, the next step is to flow in these H1 and H2 amplifiers. So this is really where the HCR system comes into play. These hairpins are engineered oligos, and in this dormant state, they don't interact with each other, they don't interact with the sample. But when they come into contact with their corresponding initiator sequences, they then unfold. So the H1 will hybridize to the I1 sequence, and in turn, this allows the H1 to unfold and bind to the H2 sequence, and so on and so forth. So this is how we get a self-assembling amplification polymer. This reaction occurs in mild conditions and is very easy on your sample. So something that we like to highlight are the relative sizes of the various molecules in your sample. These boxes are drawn to scale. So on average, your typical protein will be about 50 kilodaltons or so. Your typical antibody about 150. And where this is important is if you look at other RNA-ish amplifiers in use with other techniques out there, these strands are actually quite large, on the order of 300 or more kilodaltons. And it's because of this that they require enzyme digestion and protease treatment to otherwise break up your sample and allow permeabilization of those reagents. However, HCR hairpin amplifiers are only about 1.5 kilodaltons, and this is the largest component of the hairpins and the probes. So it's because of this feature that HCR does not require protease or enzyme digestion, and there's no substitute either. So there's nothing special that we do instead of protease. It just does not require that process. This is an example of how this is important for ISH workflows. On the left, there is a image using a different ISH product that does require that enzyme digestion. And as you can see, there can be the risk of tissue damage. And with the associated loss in morphological information, you can also get loss of signal as well. On the other hand, HCR Pro, RNA-ish, does not require that otherwise damaging protease treatment. So these are some of the features of HCR Pro kind of in a summary. So we do enable up to 2-plex RNA and N-plex protein detection. So you can detect up to 2 RNA targets and as many protein targets as you'd like. This process is always protease-free. Again, there's no fine print to that. What is nice about this process when used with protein co-detection is it's really plug and play. So you can use any IF or IHC assay that you're already using and combine it seamlessly with HCR Pro whilst on the Leica BondRx. The other features is this process has native support fluorescence. So what's nice is you can actually use the same kit, just swap out the chromogen for a fluorophore. These protocols are DNase compatible, the buffers are formamide free, and for clinical grade workflows, we have a dual channel validation service. So using HCR Pro on the Bond, there's flexible protocol programming. So they will come preloaded, but you can also adjust parameters as needed. The process is readily compatible with N-plex IHC or IF or isPLA co-detection with DaVinci. You can use any HRP or AP-driven chromogen or fluorophore. What's nice is that, again, it's the same kit. You just have to swap out the chromogen or fluorophore for the one of your choice. You can get down to a 9-hour assay turnaround time with a full load of slides. We have comprehensive setup guides, so everything you need to know about running HCR on your BondRx. And we do have free starter kits for new users, which we'll get into a little bit later. So as mentioned before, the HCR Pro platform can be combined seamlessly with not only DaVinci's products, but also just any other third-party IHC or IF assay that you may already be using. So this is an example of that co-detection. This is polar 2A RNA and PDL1 protein done on the same tissue on the bond. And this is just another example of that as well. So this is SLC17A7, the RNA target, and VGLUT1, the protein target. And again, as mentioned before, using fluorescence instead of chromogenic is seamless. You just have to swap out that reporter. Any TSA-based, HRP or AP-based reporter can be used. In this image, you can see our membrane stain. So this is a primary that we offer for users to do cell segmentation, and even for users who don't necessarily have a co-detection workflow in mind. This is a nice example of how they can integrate protein detection with their RNA detection. Another example of fluorescence, this is using a mouse tissue array, clockwise from the top left. This is mouse pancreas, lung, liver, and colon. So some testimonials from a lot of our users. We've greatly expanded our user base in the last few years. This one scientist at Neurocrine, he noted that in the past, projects took quite a while to optimize, but with HCR Pro, with the ease of use of the protocol and the reagents, validation time has been taken from 2 to 3 weeks down to less than a week, and they can quite work on final studies much quicker. And this one scientist at Strand, we have a pathologist who's really uneasy about endorsing new technology, but results of HCR Pro has impressed him with the clarity of staining and the integrity preservation of the tissue. So with the feature of HCR not requiring protease and the other features of the protocol, we're proud to lead the industry in clinical grade performance. That dual channel validation service I mentioned earlier can be a way to help validate your probes. The turnaround time is quite quick. So with a turnaround time of 8 or 9 hours, that leaves users the possibility of running 2 assays in a day, which can really speed up your turnaround for your projects. And we also have the lowest per sample cost. We don't charge any custom design fees for probes. So any target that you wish to detect for, we can easily integrate into your kits, and it's a flat kit price for each target that you order. We've collaborated with a lot of different partners, including DaVinci. So this is actually a poster that we co-presented with them at SITC in Houston last year, integrating the HCR Pro platform with the DaVinci PLA platform. So, like I mentioned before, for new users, we offer free starter kits. And what this will look like for them is a species-specific positive and negative control probe set. It'll include that membrane stain I mentioned earlier, just to try out IHC co-detection if you wish to see it. It does include control slides as well for your choice of human or mouse, but we encourage users to use their own tissues as well to just not only make sure the protocol is working as expected, but compare as needed to the control slides for RNA preservation. And it includes all the reagents you need up into the chromogen or fluorophore. So again, this is something that you can pick yourself, your preferences, as long as it's AP or HRP driven, it can be used. That is all from us. Again, thank you all for coming, and we're looking forward to getting any interested users started with HCR Pro. I'd like to introduce now Sara from DaVinci for their platform.
Thanks, Joseph and Kendra, for your great presentations. I'll now finish up with the DaVinci's in situ proximity ligation assays and how we were able to integrate that with the molecular instruments HCR Pro RNA FISH on the BondRx autostainer. So taking a step back, why is precision medicine such an interesting field to be working in? So precision medicine is an emerging field that seeks to understand the individual, be that in their biology, in their health and disease. Understanding the individual allows us to improve upon prognostics, diagnostics, treatment, and prediction, and then move towards preventing disease and deaths in a way that conventional medicine cannot do. Therefore, we will hopefully have improved patient outcomes and move towards revolutionizing the healthcare system towards a more sustainable model. So why should we look at proteins? As opposed to DNA and mRNA, proteins drive almost all of the biological processes within our bodies, and they're also dynamic. So in order to find the right treatment, i.e. drug, we need to look at proteins as they're most often targeted by those drugs. To treat the right patient, it's been shown that proteomic studies are required for the best patient stratification. And to treat the patient at the right time, as I mentioned, proteins are dynamic, which means they carry time information. Months before symptoms of a disease are seen, the disease can be reflected within the protein profile. There was a study shown that on day 0, it was possible to predict whether or not the patient would succumb to COVID, have severe symptoms of COVID, or minor symptoms of COVID by day 30. So in this sense, proteins are more actionable than DNA and mRNA. It's important to mention that, of course, genomics and transcriptomics have been very high throughput for quite many years now, but recently proteomics has been catching up in this sense. And in order to, get a better look at precision medicine, there are spatial biology technologies that are kind of constantly evolving and improving in the field. So transcriptomics gives us a snapshot of transcriptional activity within a tissue at a certain time. So this gives us spatial context. However, from this, we lack protein expression and protein functional information. Proteomics allows us to analyze protein expression and the spatial context of those proteins. However, this doesn't give us any protein function information. And then interactomics, which is where Da Vinci comes in, allows you to study and visualize protein interactions with the addition of expression and contextual information, which allows you to study more complex molecular processes that you can't otherwise. So our solution to this is the in situ proximity ligation assay. So if we take these two cells here on the screen, imagine that we've zoomed in on a tissue sample. You can see proteins throughout these two cells in the colored shapes, but also you can see proteins on the membranes of these cells. And here we are identifying those interactions with the red stars. And these are the interactions that the in situ proximity ligation assay can capture. So that's within subcellular compartments of cells and also between cells, as our resolution is around 40 nanometers or less. Now, to give you an idea of the workflow, this technology can be used in any laboratory that performs IHC or IF staining. And of course, if you have a Leica BondRx, the automated versions of these workflows is usable. Let's take the example of a PD1, PD-L1 assay. We do standard slide preparation, tissue slide preparation, so baking, dewaxing, and antigen retrieval. We then add our two primary antibodies that are specific for our targets. We then follow this by adding Navini bodies, which are secondary antibodies that are conjugated to oligonucleotides. So here's where the proximity comes in. If our two proteins are within 40 nanometers or less of each other, these two oligonucleotides can undergo a series of enzymatic reactions between steps 3 and 4 that results in a signal that is detectable by microscopy. If those proteins are not within 40 nanometers of each other, there will be no reaction and there will be no signal. So you can see in step 3, we have a circle formation. This undergoes a rolling circle amplification, which ends with a DNA bundle. And this is where we detect our signal. So you can see here on step 4, we have fluorophores that are bound. And there are many, many fluorophores bound to this DNA. So as opposed to IF, where you don't have that amplification, in situ proximity ligation assay gives you an amplified signal. So this can make it easier to detect low abundant interactions or proteins. This signal is visualized on a standard microscope or scanner, and the signal is also quantifiable using various methods. So you might ask, why not just use immunofluorescence to identify these interactions? Well, we can see here a few possible realities for proteins. Proteins may be nearby to each other but not interacting, or nearby to each other and not interacting. And they also may be distant. So when it comes to the two distant proteins, when they are greater than 250 nanometers in distance, you will have two resolvable signals, one for each of the proteins. So you understand by this information that the proteins are not interacting because they're far apart. When they are nearby, within 250 nanometers, due to the diffraction limit of microscopy, this looks as if it's one signal. So from this, you don't understand if they're interacting or just close by and not interacting. So in order to avoid false positives, isPLA, in situ proximity ligation assay, gives a much better resolution. So what can the technology be used for? There are 4 main protein aspects that can be investigated. but this is not limited. So we can look at protein-protein interactions, post-translational modifications, single protein localization, and antibody validation. So when I talk about single protein localization, what I mean here is using the amplification of the technology to then amplify signals to detect targets that may be low in abundance. So maybe there's a protein that you find difficult to see with IF. You can use two antibodies specific to different epitopes of the same protein and use the amplification step and visualization to potentially target that protein. We've also recently had customers who've tried to use isPLA for antibody validation, and they found it to be more specific than conventional IHC validated antibodies. And then around in the orange circles, we have a potential research context. Again, this is not limited. We probably haven't thought of all possible contexts. So we can use it to understand basic biology. For example, we can look at cell signaling cascades. We can evaluate drug targets, understand the response to treatments. We can, very importantly, use it for patient stratification, understand disease progression, and also validate biomarkers. So here's a case study, actually, where the researchers aim to test the immune checkpoint therapy response predictive capacity of the PD-1, PD-L1 interaction. They wanted to then compare this to the performance of the current clinical standard of PD-L1 tumor proportion scoring. So the current PD-1, PD-L1 scoring is done using IHC. What they found was that the PD-1, PD-L1 isPLA interaction is indeed a predictive biomarker of immune checkpoint therapy success in non-small cell lung cancer. So high levels of PD-1, PD-L1 interaction was associated with improved survival post-immune checkpoint therapy. Also, using a Cox regression model, including histology, stage, sex, and smoking history, the PD-1, PD-L1 interaction signal was the only variable that was associated with survival. So this highlights how we might need to look beyond just protein expression. As I mentioned, this was compared to just PD-L1 IHC, and how we can improve our biomarkers going forward with regard to patient treatments and outcomes. So we found that our technology is very integratable with existing workflows, be that automation, multiplex IF systems, and also technologies that look at other molecules like RNA. So of course, today we'll be focusing on the Leica Bond instrumentation, Leica Bond RX instrument, and also the HCR Pro RNA fish from Leica Instruments. So here's our Navini Bond RX automated workflow. You can load up to 30 samples at once, so the throughput is quite significant compared to a manual workflow. All of the tissue preparation is performed within the instrument, so that's baking, deparaffинization, and antigen retrieval. It's important to note, however, that on the Bond RX system, the antigen retrieval is automatically set to buffer ER2 for 100 degrees Celsius for 40 minutes. But every time you add a new pair of antibodies, so if you use a flexible kit, you would use your own antibodies, you need to optimize the antigen retrieval in a way that's best for those antibodies because it can vary. The only manual step really in this whole workflow is inserting the slides, preparing the Navinci PLA reagents, and also taking the slides out at the end. So a run is about 9.5 to 12 hours in total. So you can run it according to a way that works best for you, whether that be starting overnight or starting in the morning and taking the slides out at the end of the day. Again, the readout is bright field or fluorescence, depending on the kit that you use. So this is completely up to you. Comparing a manual to automated workflow, 2 days are required for a manual workflow, and we have to split it across 2 days because it's quite a longer process. So day 1 is usually shorter than day 2 because we have the enzyme required steps on day 2, so they're a bit more time consuming. The throughput is of course lower because we are relying on people to do the reactions, so there's a limitation there. And also then between different people, you can have a variation in the workflow because people do things differently. So you have a variation also between labs. Using the automated Navini Bond RX system, the assay only requires 1 day with very limited hands-on time. And of course, then the throughput increases up to 100 to 150 slides per week, which is quite a significant increase for manual workflow. Also, very importantly, the workflow becomes robust and standardized due to the system performing the staining in the same way every single time. So as we can see here, we're comparing a manual run with a Navini Bond RX run using the Navini Bright assay. We have here PD1, PDL1, CD8, MHC1, and ZAP70 LAT assays, staining normal human tonsil tissue. Antibody concentrations were kept the same throughout all of these assays. And you can see that the assay is very robust and strong between manual and automated. So there is hardly any variability between them. So yeah, it's highlighting the ability of the Navini Bright Bond RX kit to detect different interactions in a way that we see is the same standard as with manual. We also did an experiment that was published as a poster at SITC last year with ProPath to demonstrate this reproducibility across 9 human Hodgkin's lymphoma tissue sections stained with PD1, PDL1. We can see across all of the tissues the same localization of the staining and same staining pattern, which highlights again how robust the staining is across different runs. So now we move into the integrated workflow. So to combine our Navini system with the molecular instruments HCR Pro RNA FISH, we use the Navini Bright HRP kit. And we also used TSA dye detection, which also gives another layer of amplification. This is also, with some optimization, it's possible to multiplex the RNA FISH, which is an added bonus, which we can talk about in a couple of slides. So to integrate them on the Leica Bond RX system, you really follow the same workflows that we've both described. All of the steps are run within the instrument, so including slide preparation. So again, it's the same case of just inserting the slides, diluting the reagents, and then taking the slides out at the end. So we go through a seamless process between the isPLA assay and the HCR Pro RNA FISH. And if you would like to do multiplex RNA FISH, you can repeat steps 7 to 11. Again, this requires some optimization. So to test whether or not these 2 different technologies were compatible, we started with a PD1, PDL1 interaction with a 1-plex HCR RNA Pro FISH, HCR Pro RNA FISH assay. We used PPIB as it's a very abundant marker for RNA. So we can see here, this is a tonsil germinal center where we expect a lot of PD1, PDL1 signal. We can see in the middle image, we can see PPIB RNA across the tissue, and then we can see the merge of the 2. So by running the isPLA first, followed by the HCR Pro RNA FISH, we can combine the 2 technologies successfully. We then sought to try a 2-plex HCR Pro RNA FISH readout. And for this, we used PD1 and PDL1 RNA probes. And we can see here on the right-hand side that we have a successful integration with the additional RNA FISH plex. So this integration provides new opportunities to study complex molecular interactions, but also in the future can be used for tracking how does RNA move to protein, studies that we haven't really been able to do looking at the same tissue at the same time. So this is very, very exciting. And as Joseph showed our joint poster, I would really urge you to take a look. So that was a really great study that showed the future possibilities between these two technologies. From Da Vinci, coming soon, we're very excited to be launching our first multiplex interaction. So this will allow you to look at 2 protein interactions and 1 single target interaction within the same assay on the same tissue at the same time. So keep your eyes peeled for that coming out soon. We're also very excited to share with you here our first ever 9-plex isPLA prototype. This is allowing us to look at 9 interactions at the same time. And we truly believe that this is the future for protein interaction detection. And just to wrap up finally, we are a global presence despite us being a small company here in Uppsala, Sweden. We did only launch in 2020. However, we are already in 17 distributor markets, and we do have direct sales worldwide. So if you'd like to know more about the technology or understand which is your local distributor, please don't hesitate to get in contact with us, and we can provide you with more information or the correct contacts. Thanks so much for listening today. We hope that you learned something new.
I'd like to thank our speakers for their presentations today. We'll now jump into the live Q&A portion. If you do have any questions you'd like to ask, please go ahead and do so in that Q&A box. We'll get to as many as we can as we have time for. So let's go into the first question that we have. I believe this is for, I think this is for Joseph. No, this is for Kendra. How many slides can be run on the Bond RX? Can multiple protocols be run simultaneously?
Yes, so 30 slides can be run on the Bond RX at a time, and then up to 3 protocols can be run simultaneously. So each slide staining assembly holds a tray of 10 slides, and then each of those is can be its own protocol.
Right. Next one here. This one is for Joseph. What are the main advantages of the HCR platform compared to other ISH techniques?
Yeah, there's a few key ones. That big one is that non-need for protease. So that's always been intrinsic to the platform. So that allows you to preserve your tissue morphology, more easily integrate protein co-detection with that protocol. In addition, the ordering process is quite straightforward. So again, it's a flat price for custom probe designs. So for any target that you're looking to do, even if it's an unusual or unique one, it'll be cost effective for you to do it with HCR. And finally, the onboarding process is quite quick. So we offer free starter kits. The protocol is now preloaded on the Leica Bond RX. So setup is quite straightforward for new users.
Thank you. This next one will go to Sara. Can the isPLA be used on frozen or FFPE tissues?
Yeah, so our assay can be used on both types of sample, but it's good to test first, obviously, because we found that some antibodies don't work as well on fresh frozen, for example. So definitely can be used on both, but definitely do some optimizations and tests first before committing to a large number of samples.
All right, this one I believe is for Joseph. What is the difference between is that one, I believe, right? Yeah, what is the difference between the regular PLA and your isPLA?
I think this is for Da Vinci.
That's
actually.
mine. I think.
Oh, I apologize.
Yeah,
Yeah, sorry.
I can take it. Yeah, so our isPLA has been shown to be more sensitive than other PLA. And we actually do have there's a neuroscience paper that has shown this, so we can happily forward that on to you. But also we've branched out and we've optimized our kits to be specific for tissue and for cell, because we can't treat them the same way, these two sample types. So we've got optimized kits for each of those. So that's why you should use our isPLA.
All right, this one was, I believe, for you, Joycelyn. My understanding is competitors have gone protease free too. How does HCR compare with that?
Yeah, so we can't speak to the development of competing technology, but again, HCR has always been protease free from the start, so it's never needed it, even while it was still being developed at Caltech. So we never had to compromise to implement that feature into the platform. It's always been part of it. Protocol has been designed from the ground up, knowing that it's not a requirement. So the effects downstream, again, preserved tissue morphology, you don't need a different protocol or product. Any probe or assay you do will have that feature just ready to go.
All right. This one, I believe, is for Kendra. What software updates are required to run the new preloaded protocol?
Sorry. As far as the software version, you would need to be on 7.0. And then as far as the BondRx data definitions, that was 38 for the Navini Bright chromogenic, 40 for the Navini Plex fluorescent, and then 41 for the HCR Pro RNA-ish.
All right. For Joseph, what is the cost of a custom probe kit and the turnaround time?
Yeah. So the cost of a custom probe kit is $1,500, and that includes probe design. It includes the probe itself, as well as the detection reagents. It's all inclusive. The only thing that you need to provide from your end would be the chromogen or fluorophore that you wish to detect the target with. And the turnaround time is 15 business days from the order submission. And again, that $1,500, 15 business days is for any target that you can think of.
All right. Let's do for Sara. How long does it take to perform the molecular instruments and Da Vinci assays together? Which is performed first?
Yeah, so we run the isPLA assay first, and then follow that by with the RNA fish HCR Pro RNA fish protocol, and they take around. So let's say our assays are around 9 hours, plus the molecular instruments in maybe more or less the same. So it's definitely an overnight run. It's not something you would start in the morning. You definitely put it in overnight and then take the samples out the next day. But it also depends if you are doing some optimizations that can make the time vary. And if you're trying to do 2-plex RNA fish, of course, you're just going to add more time.
All right. Let's see. Why should we use HCR Pro instead of LSM or LS Multiomics from Biotechnology? I believe is the way you pronounce that. You can correct me if I'm wrong. I'm not exactly.
So again, yeah. So I mean, again, can't speak exactly to the competitor side, but from our side, HCR Pro offers that higher sensitivity, a faster turnaround time on the assay itself, about half the price per slide. Again, we don't charge any custom design fees for probe design or synthesis. And our flexibility with probe design extends to the fact that we can also share the probe binding sequences with you, no extra charge. So if that's something that your team wants to review before ordering, we can totally do that as well.
I believe this one is for Sara. What readouts are available?
Yeah, so you have the luxurious choice between chromogenic readout and fluorescent readout. Of course, depends what you're comfortable with, but both assays are performed in the same way. The only difference is the detection steps. So you can also feel free to try something new, perhaps.
All right. Can you please describe the steps that occur between the IHC and RNA fish dual protocol? Don't know who would like to take that one.
Maybe they meant isPLA. Let's assume that they meant isPLA on the integration. Yeah, so it's really just a few bond wash steps, nothing particularly special. And then there is a step there to remove the HRP that we've used to detect the isPLA signal with. And then we really just jump straight into the beginning of the HCR Pro RNA fish protocol. That's really it.
Okay. For Joseph, can we request the probe binding sequences after design is complete?
Yes. So that's something that you can do, no problem. No extra fee for doing so. You can just request it via email. We're happy to share those with you during the probe design process.
All right. Is isPLA compatible with Alexa FLUOR secondary antibodies?
So our fluorescent kits, they already come with the detection mixtures. So it's not, with an if you have a secondary detection IF, then you would need a secondary fluorophore Alexa Fluor dye, for example. But in this case, you have everything that you need in the assay, in the kit that you receive. So there's no need to buy anything extra.
HCR question, is it possible for you to create a probe for 2030 base pairs?
Yeah, so that is definitely a shorter target. For that one, you are more than welcome to submit this to our team, and then we can communicate with you to see what's possible.
For Sara, I believe, is there support for the analysis of the images?
Yep. So we can provide support and guidance on how you could best do image analysis for our signal types. So please we can get in contact with you. I think we must have your information. Just we'll drop you an email and we'll see how we can help.
Okay, we've got time for a few more here. I believe this one might be for Joseph. Is there software to measure, quantify each probe?
Yeah, so we actually do have an official module with Indica Labs and the Halo software, as well as VisioFarm. So if you already have those modules or that software, you can readily use that with your HCR assays.
For Sara, can I use my own primary antibodies?
Yeah, absolutely. So we have targeted kits, PD1, PDL1, and CD8 MHC1. But we also have the Navini Flex kits. So this is bright field and a fluorescent, where you can use your own primary antibodies to look at whatever you're interested in, be that protein interaction or single protein.
One more for Sara. What optimization is required?
Yeah, it's a great question. So follows quite nicely from the previous one. If you're going to use your own primary antibodies, whereas when you buy our kit, a targeted kit will tell you, exactly how to dilute the antibodies, which concentrations, we've done all that testing. So you kind of have to do a bit of optimization there. And we suggest that you first test the primary antibodies in IHC. And when you find a concentration that's working, you can use that for your first isPLA assay for those two antibodies, and then you can go from there. You'll figure out the right balance. And if you do need support with that, you can always contact us for help.
We've got time for one more, and I think this one is for Joseph. Can you come on site to help with setup?
Yeah. So with the recent launch of the pre-programmed protocol on your bond, that does make setup a lot easier. Our setup guide as well kind of walks you through not only running your assay, but also walking you through the starter kit even. That being said, if you do need additional help, we're open to coming on site. We are a small team, so we can't promise we'll be able to get there immediately. But again, if you need extra help, do feel free to reach out and we'll see what we can do.
All right. I'd like to thank our speakers again for their time and research. We'd also like to thank LabRoots and our sponsor, Leica Biosystems, Molecular Instruments, and DaVinci for underrating today's educational webcast. Before we go, I would like to thank the audience as well for joining us and for the questions. Any questions that we didn't have time for today or that get submitted during the on-demand, they'll be addressed by the speakers via the contact information you provided when you registered. Like I mentioned, this can be viewed on demand, and LabRoots will alert you when it's available for replay. And we do encourage you to share that email with your colleagues that might have missed today's event. So until next time, thank you very much, everyone, and goodbye.